pushing record. Okay, so can you see all my little gray background slides?
Yes.
Yeah.
Yes? Okay, good. All right. So when we look at this slide right here on the cell cycle, you can see the interphase is where the cell spends most of its lifetime. And it's approximately 10%, 90%. It varies a little bit, you know, cell type to cell type, but mitosis is quick. And mitosis is simply the process of dividing the nucleus of a cell, a eukaryotic cell, into two nuclei, and cytokinesis is dividing the cytoplasm and the plasma membrane around those two new nuclei, so that then we have two cells. And G1, S, and G2 comprise most of the time of a cell's particular life within a cell cycle. And G1 and G2, the cell is growing, that's why it's G. It's manufacturing proteins, it's creating cell membrane, it's bringing nutrients in and waste out. And that means that, thank you, sweetie, go find daddy. That means that, you know, it's not static. The cell isn't just the way it started at you know, minute 0 until the time that it divides. But right in here, S phase refers to when the genetic material, the chromosomes, are replicated. And as soon as S phase occurs, then the rest of interphase, the cell is made of, you know, it's getting bigger until it you know, reaches the G2 checkpoint and passes into mitosis. And, you know, essentially, we need the DNA to be replicated, because if a cell divides without it, then these cells won't have the same amount of DNA as the parent cell. And as you can imagine, that could present problems for what proteins need to be made later down the road. And so this is for all somatic cells, non-gamete forming cells. And then the separate process for the cells that are going to produce gametes, so reproductive cells that make gametes, those perform meiosis, so a variation of nuclear division. And if I open up the PowerPoint for chapter 9, it's just gonna pop in over here. I believe I have a Zoom recording that goes through a significant chunk of this. Does that sound right? In fact, there's 2 of them. Yeah?
Yeah.
Okay. So I don't need to cover it all again. What I want to show you here is that, you know, the focus of this chapter, the second half is on this process, these 2 processes combined, transcription and translation, where transcription is making an expendable copy of DNA that can leave the nucleus, which is RNA, and then outside of the nucleus, whether it's in the cytoplasm, on the surface of the nucleus that has ribosomes, or the rough ER, and it's rough because it's studded with embedded ribosomes, any of those places, or even in the mitochondria, there are mitochondrial ribosomes. Any of those places where there are ribosomes, the RNA can be sent and protein can be manufactured. So let me back out of that slide. And so then the first set of slides that I went over that's in the Zoom recording walk you through replication and mutation, how DNA can be damaged. And your DNA is getting damaged hundreds of times a day in cells in your body, you know, different cells, not all at the same time. And it's just as frequently getting repaired. And the reason that you care about those repair mechanisms is that if the DNA in somatic cells is mutated, and then that cell replicates its DNA and divides, then those 2 new cells that have split the DNA have replicated mutated DNA. And if you have enough mutated DNA in cells, then what process might not be followed properly? What do the DNA code for?
Protein.
Protein, right, yes. Thank you.
I forgot I was on.
It's okay. No, I want you to answer.
Cool.
So in a somatic cell, we've got DNA replication, and then the cell divides, and you know, before, during, and after that whole time, different pieces of chromosomes are being you know, unzipped and read, and RNA is being made, and the RNA is being sent to its destination, and protein is being made. But if those cells have mutated DNA that's not the original DNA that the cells in that organism started with, then it can make proteins that don't function, or incomplete proteins, or a particular protein no more that was coded for before. It can make proteins that don't tell the cell to stop dividing when there's not enough space. It can make proteins that attach to genes that turn them on, so too much protein is made. And or that tell, so you can end up with diseases with enough, you know, mutated DNA where your proteins become dysfunctional, right? And what about if you have proteins that are mutated that turn the cell cycle on and to stay on, or tell the cell cycle to go faster? Then what happens to, yeah, cancer? Cancer is essentially uncontrolled cell growth. And the reason that humans and other organisms develop cancer, uncontrolled cell growth, is an accumulation of unrepaired DNA mutation in somatic cells. None of it is heritable, at least in somatic cells. You don't pass it on to your kids, you don't pass it on to your dog, you don't pass it on anywhere. It's just errors in your cells. And we have a lot of repair mechanisms that take care of it in the cell. And there was a video that highlights this. There's a link down here on this slide, slide 22. And there's also a video about it in my my videos, my curated videos list for chapter 9. So I just wanted you to be thinking about that. So mutations in somatic cells only affect that living eukaryotic organism and don't carry on with it when it passes. So where do we get mutation that can cause problems? There. Okay. So what you see here is a preview for the lab that's due the middle of next week. And like I was saying, the lab lags behind just a bit. But if I click on what this assignment is, there's a website on the lab page here. And if I click on it and show you, you walk through an explanation of the cell cycle and the checkpoints. So it's totally gonna nail what you were missing. Like, oh, that's it, right? So I have some of it in my recordings in the PowerPoint. But this sheet is what you would complete and see how it's just gonna walk you through everything with cell cycle regulation. That's half of the lab, okay? And then the other half of the lab, if I click on lab module 8 and I open it and put it on my other screen, let me pull it over for you. Okay, so if I scroll through, it reminds you, hey, your first part is to do the cell cycle. And then it walks you through identifying cells in mitosis and meiosis and matching the phases of mitosis with the description over here. And then you actually get to practice making model chromosomes yourself. And then what you do is then you try to identify again here. By the way, I would do this on the computer, not on a fuzzy printed out version of this, right? So you can print it if you want and then write on it, but if you're gonna actually look at the images, do it while it's on a computer screen because you'll actually see things, okay? And then, so you practice down here, and then you get to walk through like what happens in meiosis and practice drawing it yourselves. That's what all of those are. And then walk through what happens with independent assortment and crossing over, okay? And that's the lab. And like I said, it's coming after the multiple choice test, but you don't have to, you're not actually having to move stuff around on the exam. It's the practical application of this is going to be answering some questions that are on the lab quiz that are next week. So I'm going to minimize this and I'm going to come back over here and show you in the video section where it says Zoom sessions, that Canvas webpage that I've created. And as I scroll down, sorry if I'm making anybody ill, it makes me blink to scroll really fast. So mitosis and meiosis is where the cell cycle and the checkpoints are that I was talking about. And then I'm looking, I thought I had a recording for, oh, here we go. Do you see this one that says Chapter 9, Transcription Translation Part 2 with mitosis lab help at the end? I'm trying to, these, my Wi-Fi went down, and so I had to get everybody to log back in. That's why it's split. And so then we went ahead and wrapped up. So I'm going to come in and have it skip ahead. I go over the homework, one of the learning assignments here. That's on transcription and translation, and a lot of it. And then I'm just waiting for when, oh, here we go. So here's the lab. And so it looks like the last about 20 minutes. Yeah, there's about 20 minutes where I go over this particular lab for next week. So if you wanted to preview next week's lab by watching that portion of the video, it might help you a little bit with the test, besides with the lab. All right. I have to figure out where to close. There we go. So, I'm trying to look around my smaller screen to see what else I still have open down there. So if I go to this checkpoint, this is in the video, but just to overview a little bit, to answer the question just in the slightest, but now, the G1 checkpoint is looking for growth and for, you know, having resources, okay? And essentially, if everything is there and in proper order, you know, the chromosomes are complete, etc., then once the cell passes the G1 checkpoint, it tends to blow through everything else, as in it finishes the cell cycle and it divides. If we get to the G1 checkpoint and there's no go-ahead signal, then the cell goes into what's called G0, which you would see as a G with a 0 subscript, and it means it's simply not dividing. Most cells can return from G0. Some cells, if they get sent to G0, it's basically they never divide. They're in a permanent state. Most of our neurons are in G0, which is why, you know, brain damage is so hard to recover from, if it's recoverable, because of cells being in that non-dividing state. But anyway, past G1, you go through DNA replication, and to get through G2, this is the maturation promoting factor checkpoint, where we're looking for the accumulation of a protein complex that basically, once there's enough of it, it triggers the cell's passage from interphase into mitosis. And then the M checkpoint happens during metaphase, when all the cells are lined up, not the cells, excuse me, that was a bad misspeak. When all the chromosomes that have been replicated are lined up at the center, which is referred to as the metaphase plate, then the cell gets to check and make sure that all the microtubules that are going to separate the sister chromatids to opposite ends of the cell are actually attached. Because if they're not, then you end up with something called non-disjunction, which is chromosomes that don't separate, and you end up with too much DNA of a particular chromosome in one of the new copies of the cell and not enough in the other one. If it's a somatic cell, it can result in a non-functional cell because of certain proteins that are needed, or a cell that doesn't behave properly. If it happens in meiosis, where we don't pass the M checkpoint properly and the cell still divides, that's when you get things that are often lethal chromosome aberrations, when like chromosomes 1 through 7, the bigger chromosomes, have non-disjunction and they don't split up properly. It's often lethal in that the embryo can no longer develop and divide, and it doesn't produce, you know, a viable organism. When we talk about chromosomes that are smaller than that, then some of those just lead to, you know, big developmental challenges. And we don't have every genetic illness or genetic, I'm searching for the right genetic abnormality. We don't have all of those mapped out yet. We don't know every chromosome and where all of those come from and exactly what happens. And an example I could give you is my 14-year-old has a visual impairment called optic nerve hypoplasia, and she was born with it. It's not inherited, excuse me, but it's a new mutation that they think may have a genetic component that happens randomly and may be more common in like in a heritable fashion, but they can't say for sure that it's inherited yet. So there's no chromosome to point out and to, you know, hold responsible for her genetic condition. It's just something that she was born with. And then there are illnesses or things that are different that we understand very well. I have a brother with Klinefelter, and I don't know if you know what that is, but when the egg formed that was fertilized by a sperm that made him, that egg ended up with 2 X chromosomes. And so when that egg was fertilized with the sperm, it ended up XXY. And that's a non-fatal genetic but not inherited condition in humans, in that it's not something that either of our parents had that was passed on, and it's not something that he could then pass on, but it is in the cells that were formed during the process of meiosis, that nuclear division. So it's that kind of genetic, just not inherited. So in case you were thinking that genetic and inherited were synonymous. Mm-mm. And I'm sure if you took a genetics class, if this is like making, you know, your hair stand on end and you're getting all excited and interested, you know, take some genetics classes going down the road. Maybe you'll have somebody as great as my Dr. Evans was when I was in college. Didn't quite dissuade me from studying marine ecology, but it was pretty close. Okay, so any other questions before I spend like 20-30 minutes doing some practice exam questions?
Nope.
Nope. That's what you're all here for?
Pretty much.
Oh, that's all right.
Not that the review isn't appreciated.
Right.
Well, hopefully that helped you kind of pull the big ideas back to the front of your brains and out of the archives again. All right, so on mine, I have to click on assignments. I have to keep things in all these little folders so that that it will show up the way it's supposed to in your grade book. All right, so it gave us a subset of questions that we're just going to work our way through. Okay, and I think this is one that everybody gets because I have some tough ones that I just show you. So if you can see, these cells are numbered 1, 2, 3, 4, 5, 6, 7, 8, 9 through 13, and 14 through 18. And this one says cell number 1, and it's mitosis and onion cells. So if I was going to choose, what does seeing all those little X's down the middle tell you that the cell is doing?
Metaphase.
Metaphase. Okay, yep. And these are sister chromatids that are, you know, together there. That's why you see the X's, but they're not, and you know it's mitosis, so you don't expect them to see like double X's together. Okay, cell number 2. Do you see these little hashed marks? That's the circle, but it's not complete. And then you see the X's that are all kind of loose. So interphase, anaphase, telophase, or prophase?
Prophase.
So when does the nuclear membrane come apart? When does it disintegrate? It has to get out of the way in order for the chromosomes to get it to attach to the microtubules and get moved around. So it has to happen first. And you can see that they're X's, so you know that the DNA is replicated. So we're not going to choose interphase, right? So anaphase, telophase, prophase.
Prophase.
You sound like prophase, please.
Well, that one.
It is prophase.
Yeah.
Okay. And remember that I mentioned the last time, what might you want to do if you're capable? Have a split screen, or what? Have a second screen, and so then really quickly you can pull up something like this and be like, aha, I see them here, and this shows me that it's coming apart, so prophase, right? Or if you drew yourself a diagram and had it all together, that might be really helpful.
Okay.
All right, so cell number 3, do you see onion cells? So it's a plant. So it's not a line, it's these little segments. And then you see 2 nuclei here, and you see little squiggles, but they're not X's.
Telophase.
So, and notice the nucleus isn't completely reformed. So I'm going to show telophase, and then this thing forming down the middle is called a cell plate. It's showing you that cytokinesis is happening, right?
Yep.
And then let's look at cell number 4. It's a single cell with no X's, and this nuclear envelope is solid.
Interphase.
Yep. Okay, and cell number 9, this guy over here, anaphase, and do you see how they look like they're halves and they're being pulled apart, and it's like, you can see my hand still, right?
Correct.
So here was my X, and now as soon as the microtubules pull on it, they're getting pulled apart because this is where the string is attached.
Yep.
Okay. All right, so anaphase, great. All right, number 2, it says identify the process and the sequence. So you have 3 images, and this hint comes right off of one of your learning assignments. And so you need to make it a little bit smaller so we can see. So it says, what's the process? So you have lovely things to choose from. Is what's happening transcription? Is it replication? Is it translation? Is it
Translation.
termination? Okay, so it says identify the process and the sequence. So the process is translation. We are making a polypeptide chain. And then image 1, image 2, image 3. Well, let's look at that. Let's think about what each one is showing. So image 3, you've got this thing that says a release factor, and you've got a chain of amino acids. So what is that, the beginning, the middle, or the end of translation?
The end.
The end, right? And then look here, you have UAC on the anticodon and AUG right here, and you have tRNA molecules that are actually carrying amino acids coming in. So beginning, middle, or end?
Beginning.
It's the beginning because methionine, the start codon, those are the clues. Okay, and now you see 2 are attached within the ribosomal complex at the same time. You see that the RNA has moved partway through. You see a chain that's growing. You see some tRNA molecules that don't have amino acids attached, and you see one coming in. So we've used up end, we've used up beginning. This has to be the middle, okay? So the beginning was image 1. So let's look at image 1. And so what would you say is the right term for the beginning?
Initiation.
Okay, good. And image 2, that's the middle. And what shows that something is in the middle? What's happening to this chain?
Elongation.
It's elongation.
Elongation, I mean.
Yep, because it's getting longer. And then what term shows that we're at the
Termination.
end? Termination. Good. Okay. This one, I can't remember if it's on the a learning assignment or not, but I believe it's on a practice quiz. All right, so it's showing you just some parts of the cell cycle, not necessarily everything for sure. But when we look at this, do you see any homologous pairs, any tetrads where the replicated chromosomes are overlapping and in a set?
Nope.
No. So we're, mitosis, right? And what gives it away right here? See how the chromosomes are lined up singly or individually? Yep. That's a give. And because we only have, 5 things showing here and not a double set, we know this is mitosis. But number 5, what would you say this is?
Metaphase.
Metaphase. So label something, right? And notice we've got a choice here. You could say mitosis, you could say G1, you could say S, you could say meiosis. Careful and make sure you don't say metaphase of meiosis 1 because you don't see tetrads. It's metaphase of mitosis, okay? Number 4, what do you see happening here?
Telophase.
And how do you know it's telophase? What's this pinching in?
That's the
cleavage furrow.
Exactly. Yeah. And the nuclear membrane, is it reformed now?
Yes.
Yep.
Yep. Do you see microtubules all across the cell?
Nope.
Nope. And you see unreplicated chromosomes. So that tells you it is
Telophase.
Telophase. And we're sticking with mitosis, right? So
Yep.
we want to find telophase of mitosis. And to make sure you didn't mistakenly think it was telophase of meiosis, I made sure to just put telophase of meiosis 1 because telophase of meiosis 2 and telophase of mitosis look similar, except that you would see half the number of chromosomes. But unless you have it like all out together to compare, it can be a bit challenging. Okay, number 3. What do you see forming up here that's replicated?
I forget the term.
They're centrosomes.
Yeah.
Yeah.
Improving.
And with the microtubules coming off of them, we can call this whole thing an aster. They've replicated. You see some dark squiggles in there. Okay.
Interphase.
So yeah, and you know it's interphase because these guys, the centrosomes, replicate during G2. So for 3, we're gonna say, oh, actually, I take it back. They replicate during S because that's when the DNA is replicated and that's when the centrosomes get replicated. So we're in interphase, specifically in S phase. G2 is just hanging out and waiting for the cell, for the MPF to accumulate. All right, number 2, you can see you've got a mitotic spindle forming and the nuclear envelope is fragmenting. And you've got definitely sister chromatids in each chromosome.
Prophase.
All right, so prophase of mitosis. And then this one where the sister chromatids are being pulled apart and you see a
Anaphase.
spindle. Yep, anaphase. Good. Okay, number 4, can you see how this is 5 to 3 and then running the other way is 3 to 5?
Yes.
Okay, so down the middle, joined together, we have what part?
Nitrogenous bases.
The nitrogenous bases that are hydrogen bonded together. And this pentagon, that's a phosphate. That's a
yeah.
phosphate or a sugar? Which one?
It's the phosphate.
It's
the sugar.
That's a sugar. Okay.
And
5
50,
carbon.
5 carbon sugar.
5 carbon sugar. And actually, there's an oxygen in the ring and it has a little branch sticking out that's attached to the base. But if 5-sided shape makes you think 5 carbon sugar, that works for me. And then the little circle is the phosphate. So A is pointing to the 5-sided thing. So A is the ribose or deoxyribose?
Deoxyribose.
And it has to be deoxyribose because it's a double strand.
Got you.
Okay. And B, what do we decide that was? Yep. Okay, C, this guy?
Nitrogenous base.
Uh-huh. And then D, the whole thing together, a nitrogenous base, a deoxyribose, and a phosphate?
That'd be amino acid.
No. A nucleotide.
That was going to be my second guess.
DNA is a nucleic acid because it's a polymer of nucleotides that are joined together in the covalent bonds between the phosphate of one nucleotide and the 5 carbon sugar of the other. And then we just have matching up where the A and T and C and G can hydrogen bond with each other. Did you notice what I did here, where I tried to get you to think of what they were before you started choosing? If you had just pulled open this list, you see it's not just, oh, I only need to choose the correct one out of 4. There's what? A whole bunch of stuff relating to proteins in there. So you have to know what's going on before you just start willy-nilly choosing something. So take an extra 5 or 10 seconds with a question like this and be like, okay, do I know what these are? Do I know what this is showing? And then go to the choices. Oh, so there's a video where I walk through this, but this is looking at an RNA sequence. Can you read it and can you make the amino acid chain that comes from that? So we just start out with mRNA and we just start from where we are with this partial sequence. So I would look at the first 3 are U, 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 and I would find U, 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 and I would say, okay, it's PHE, which is phenylalanine, okay? And so I know it has to be the second or the third one, but after U, 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 I have U, C, U, and I find UCU here is SER, which is serine. And so it's phenylalanine, serine, and the next one should be TYR, which is tyrosine. And so I'm going to go here and I'm going to double check UAU, the third codon. And I want to find UAU and make sure. So here's UAU, and TYR. And if on the computer that's hard and you would like accidentally read 2 and then skip, write down the sequence and then draw through it. Okay, so like draw a line straight through between the third U and the fourth U, and then draw a line that is between here and here, and then draw another line there. Are you following me? So that you can split the mRNA into its proper codons. And so a codon is just a 3-base sequence that codes for an amino acid. And technically, it should be a 3-base sequence that is an exact set of 3s downstream from AUG, the start codon. But when you don't see AUG, you just start with the beginning. All right, now this one, it says, hey, a peptide has the partial sequence of PHE, PRO, LYS, GLY, PHE, and proline. Which of the following sequence in the coding strand of DNA could code for this peptide? So now you have to go backwards a little bit, except for one thing. If we're going to go DNA to RNA to amino acids, what does DNA have that RNA does not?
Thymine.
Thymine, correct. And what does RNA have that DNA does not?
Uracil.
Uracil, okay? So look at this first option. It's full of U's. Could it possibly be DNA?
Nope.
Okay. Look at the second option. Could it be DNA?
Yep.
Yep. Look at the third option. No U's. Could it be DNA?
Yes.
Okay. This one has U's and T's. And you might be like, oh, I should choose it, AUG. AUG is the start codon for RNA. So we're between the second and the third option. And the question is, does PHE, what is it going to be? So what does it start with? Okay. And they're both 5' to 3'. Okay. So when I take TTT, what is the RNA that's going to be made from TTT? What's opposite T in RNA? A. So then I use the codon table and I say A is for lysine. That's not it, okay? And let's make doubly sure we're wrong. CCC would code for GGG. And when I find GGG, that's glycine. That's not the second one either. So I don't think it's that one, right? So now let's see, GGG. GGG is glycine. Wait a second, what the heck is going on? And then, okay, so both of these make sense. But then look at the priming. What do we have to do? Which way is RNA read? Look up here. Which way do we read the RNA to make the protein?
5' to 3'.
We read 5'. But when I go from, oh, I'm going not far enough. When I go from 5' in DNA to making my RNA, do you know what the priming becomes? It's AAA, but it starts at 3. So I want to go to this other end, which is 3'. And what is opposite CCC and RNA? G, G, G. Oh, wait a second. GGG is still what? GGG is glycine, so that doesn't make sense. But over here, opposite GGG would be CCC. And what do you see right here? We've got proline, and then this ACT, I'm gonna laugh if I
Mhm.
had somebody mess this up on me. So this would be U, and this would be G, and this would be, no, sorry, this would be AUG. So here's my start codon, okay? So there would be methionine, okay? And then this one, CCC, would be GGG,
Yep.
which I see glycine, okay? AAA would be UUU. And so that's PHE right here. TTT would be AAA. Normally I write this all out and it's hard for me on the computer. I better start writing. We're spending a lot of time on one like this. So you know what happens if you're like gonna take it twice, right? And you feel like you're running out of time, what do? Then you you skip it and you choose another one, and you kind of does the system let you flag it and come back to it?
No.
No?
No.
Okay, so that's a flaw that I can't do anything about. Okay.
Which
A.
following sequences in the coding strand DNA?
So I'm writing them all down on a piece of paper to make sure that I get this done right. Okay, so let me highlight it. Working with this one here, GGG becomes CCC, which is proline. TCA becomes AGU, which is serine. I'm thinking that maybe I'm gonna just give everybody a bonus 2 points because I may.
You've lost me.
I'm looking
Oh,
at this
boy.
going like CCC. Because that one's glycine. I'm in, oh, you can't see me touch my screen. Ah! All these reasons to hate doing stuff on the computer. I'm coming up with that. So AAA is UUU, which is PHE, okay, right here. And then TTT, this one is AAA, which is lysine, LYS. And then UUU is what AAA becomes, which is PHE again. And then GGG becomes CCC, which is proline. So I get right here, proline, PRO, PHE, lysine. Okay. That's what I get for that one. And it's not, that's funny because that one is 1, 2, 3, 4, 5, 6, 7. And this is 1, 2, 3, 4, 5, 6. So I want to double check part number 2 again, the one that I don't have highlighted, and make sure I didn't steer you wrong because of me trying to do it just on the computer. I love paper for stuff like this. So you can see that I wrote it down, and now I'm going to take it backwards. So CCC is GGG, TTT is AAA, then opposite GGG is CCC, then I get UUU, GGG, and AAA. And so GGG is glycine, AAA is lysine, CCC is proline, and then UUU is Alanine. This is actually based on what I've just gone through. The one that I would pick, because as I go through it, I only get the amino acids that are showing up here. I have a proline, phenylalanine, glycine, and lysine. So if you get something like this, rule out the ones that couldn't possibly work, and then try to get the priming to match. And I think I might go in and trick it and make the priming opposite for the wrong one, just so that's a dead giveaway. Okay.
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 All right. Let's do one more tricky one. So mitosis, meiosis 1 or meiosis 2. You've got to know their similarities and their differences. Which process produces haploid gametes?
Meiosis.
So it's meiosis, and you get the gametes at the end of meiosis, so meiosis 2. Okay? Which process creates diploid daughter cells? So they have they're 2N. They have 2 copies of every chromosome.
Mitosis.
Mitosis, good. Now, which process creates haploid interim daughter cells? So remember, meiosis is 2 rounds of division, but in the first round, the homologous pairs separate. So that means that those daughter cells are haploid, because even though they have sister chromatids, they only have half the number of chromosomes that was in the parent cell. So meiosis 1 creates haploid interim daughter cells. Which process creates identical daughter cells, copies that are the same as the original
Mitosis.
cell? Mitosis, good. When do you see tetrads? Paired homologous chromosomes.
Oh, right. Meiosis.
Okay, but what round of meiosis? The first round of
One.
division or the
First.
second one? Yeah, the first. Okay. When do you see homologous pairs, those tetrads at the metaphase plate?
Meiosis 1.
Which process separates identical sister chromatids?
Mitosis 2.
There's no mitosis 2.
Meiosis 2, I meant to say, sorry.
Look at these two here. Separation of identical, separation of possibly recombined sister chromatids. Now, mitosis creates identical daughter cells, so we separate identical sister chromatids from those chromosomes in mitosis. And then if crossing over has occurred and some genetic information has been switched on homologous pairs, then meiosis 1 separates the homologous pairs and meiosis 2 separates possibly recombined sister chromatids. So what did I just say separates homologous pairs? The first round of division in meiosis 1. I can tell a few of you might be coming back to that one to look it over again, right?
Yes.
Possible.
Okay. All right. Number 8. Which of the following investigators was or were, if it was more than one, responsible for the following discovery? So in DNA from any species, the amount of adenine equals the amount of thymine, and the amount of guanine equals the amount of cytosine. So we're talking about A equals T and T equals G. Do you remember?
Wasn't that Griffith?
No, close.
Chargaff?
It was one person, and it was Chargaff. It's Chargaff's rule. Griffith did some fun stuff with DNA and figuring out looking at sulfur and looking at phosphorus and phosphate groups and things like that. All right. It became apparent to Watson and Crick after completion of their model that the DNA molecule could carry a vast amount of hereditary information in which of the following. So where's the coding for the proteins? Is it in the sequence of the bases, the complementary pairing? Is it in the phosphate sugar backbones, or is it in the side groups of nitrogenous bases? Well,
Complementary pairing.
complementary pairing is how we have, they figured out the complementary pairing, and that's how come we can have a double helix. But the sequence of the bases, A, G, T, G, 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 that's the sequence that contains the code.
That's right.
Makes
The
sense.
complementary pairing is how we keep DNA, what's the right word here? It's part of how we go from generation to generation, replication cycle for the replication cycle, where we keep it whole in that we minimize mutation because there's always a partner there that it should match, and the DNA polymerase can check against the partner. Hey, who's that? Your opposite you. That's, you have to be opposite that and be the right base. And so the complementary pairing helps reduce the potential impact of mutation. Okay, so in trying to determine whether DNA or protein is the genetic material, what did Hershey and Chase do? So you've got a learning assignment where you had to watch a couple of videos and write some of this information down. This might be where you want to go back to. Okay, so DNA, does it have nitrogen? And protein does not. Does protein have nitrogen? It does. So this is not correct. DNA contains sulfur where protein does not. That's inaccurate. Protein has sulfur. RNA includes ribose while DNA includes deoxyribose sugars. So that's it's true, but is that what they used?
No.
DNA contains phosphorus, but protein does not. So you have to go figure out which one did Hershey and Chase make use of, this one or that one. Okay. All right, number 11. A new DNA strand elongates only in the 5' to 3' direction. And why is that? Let me make this just a little bigger again. There we go.
Oh, DNA polymerase can only add nucleotides to the free 3' end. 3' end.
Exactly, because that's where the OH group is hanging out from. And if you've ever seen a relay race and the batons being passed, if there's no runner like with the baton out and somebody's coming, like if the baton isn't put out, then there's nothing to grab, there's no connection and the race fails. So DNA polymerase has to have a baton holder. There has to be a 3' hydroxyl group. Okay, number 12. The following structure is and functions to. So what is this thing with the 3 rings and a little bit up at the end? It's tRNA. So it's either the second or the third. And what does it do? Deliver an amino acid during translation or transcription?
Translation.
Yes, good. All right, 13. What allows DNA to replicate? This goes back a little bit to the one we were doing just a minute ago.
Would that be the complementary pairing?
Yes, that was my hint, that it goes back to that one. I didn't want to give it away back then.
No, I didn't even think of that. I was just, the other 3 didn't really make sense. I was like,
yeah, they don't.
Give it a shot.
Well, and disulfide bonding, that happens in proteins. So if you weren't really paying attention and you were like, oh, I recognize that. No, it's because you're remembering from proteins from like chapter 3 or 2. All right, number 14. If a cell were unable to produce histone proteins, what would be the likely effect? So histones are the special proteins that the DNA double helix winds around and creates nucleosomes. So it kind of, it keeps it from being all dangled around the cell. Think about what you do with a spool of thread. Why is the thread on a spool? Why do you wind kite string around something? What do you do? What do you do with an extension cord? And what if I asked you to fit, you know, X number of Christmas light strands into a certain box, but I didn't give you any way to wrap it? If you don't have histone proteins, then you'd have a big problem with being able to wind and compact the DNA and get it to stay where it's supposed to be. We have to unwind it off those histones in order to read it to make RNA, and in order also to open it for replication. Number 15, what is meant by the term anti-parallel when we're talking about DNA? Anti-parallel is referring to the sugar phosphate orientation. And that 5-carbon sugar, we label it and we go 1', 2', 3', 4', 5'. And one strand's sugars point in the opposite direction of the other strand's sugars. So the 5' to 3' direction of one strand runs counter to the 5' to 3' direction of the other strand. There is a short Amoeba Sisters video that explains the components of DNA and the anti-parallel structure in the chapter 9 videos on replication. Okay, 16, what would happen if there was no telomerase? That's an enzyme, the ASE gives it away. And we're talking about a eukaryotic cell. If we don't have telomerase, then something happens to telomeres, the repeating ends of linear chromosomes.
Would that be the high probability of becoming cancerous?
No, actually, it's a reduction in chromosome length. Telomerase creates these repeats and keeps, because of having to synthesize in the 5' to 3' direction and needing that 3' OH group, there's this little snippet at the end of a linear DNA strand on either end that can't be synthesized because of needing a 3' OH group. And so what telomerase does is it comes in brackets around that end, and it has it's a special enzyme that inserts and lengthens and protects the ends of the linear chromosomes to keep them from shortening. And one of the telltale signs of Alzheimer's, genetically speaking, is shortened chromosomes. So it's a sign of aging.
Right here.
What's that? Oh,
Not you.
Okay. Okay, no problem. Mine are watching Peppa Pig. All right. Why does the DNA double helix have a uniform diameter?
Purines pair with pyrimidine.
Okay, so yes, but which is correct? The first one or the second one?
The second one.
It's the second one, and the only way I remember it is single. It's short, but pyrimidine, the word is long. It's opposite what I would think it would be. The long word goes with single, and that's how I remember. Okay, all right. So correctly identify the timing of DNA replication in the cell cycle. So what part of the cell cycle does DNA make a copy of itself?
S.
S.
S of interphase. Make sure you don't accidentally choose S of mitosis. Because interphase is most of the cell cycle. Mitosis is when the nucleus divides. Okay. Looking at proteins, which point mutation would be most likely to have a catastrophic effect on the functioning of a protein? All right, so the point mutation is in DNA. So if I change a single DNA molecule, a single nucleotide, if I swap the nucleotide out, is anything down the strand affected? It
If you-
wouldn't be. It's just a base substitution. If I just swap it out, then I might change one amino acid, maybe. I might actually randomly get the exact same amino acid because of the substitution. Might still code for the same amino acid. Remember we were looking at that codon table a bit ago?
Yep.
And the codon table is redundant. There's 64 places, but only 20 amino acids, which means a whole bunch of the codons, even though they're different, have to code for the same amino acid, and that's called redundancy. And it minimizes the effects of mutation. So a base deletion where something's removed and everything shifts at the start of the gene,
Yeah,
at the end of the coding sequence, near the start of the sequence, but not the initiation codon. So,
it's
you
gonna
know,
be the second one.
so is it near the start of the gene or near the start of the coding sequence? We got to choose between those.
Start of the gene.
I would say for me, it's the start of the sequence that actually codes for the protein.
Yeah. They threw me
Because-
off with the, but not the inhibitor codon, or initiation codon, I mean.
Right, well, because yeah, that's just adds a little bit. That would mean that you get like a totally different protein. Where this one, a base deletion near the start of a gene, you might get no protein at all. Or it might even be not even be a part of the strand that is meaningful, because there's a lot of genetic material that doesn't actually code for an amino acid. It's just spacer stuff in there. Is that kind of strange to think about? Some of your DNA doesn't actually code for anything. It's just, you know,
Makes sense.
packing peanuts maybe to keep
I mean, they are
everything
pretty
else.
full.
Yeah, where, you know, where it belongs. Okay, number 20. What do you find between the paired complementary bases of the double helix in DNA?
I always get this one wrong. Hydrogen?
They are hydrogen bonds.
But
'Cause they're
what?
Yep, you got it. Okay. Number 21. So which mutation would not change the amino acid coded for by DNA?
Substitution?
A substitution changes it, and it may not, but for sure. So a silent mutation is a substitution mutation that does nothing.
Oh.
Okay,
There you go.
it's because it codes for the same amino acid.
Wow, I know.
Okay, number 22. We've got somebody brilliant on the other end. They know.
Ready.
All right, which of the following is not a mechanism for cells for mitigating the impacts of mutation during DNA replication? So the easy way to say that is most of these are ways a cell either prevents mutation or fixes it after it happens. And one of these isn't a way that the cell can do that. So if a cell finds nucleotide bases that are not correctly matched, like A paired with G, it'll fix it. That's mismatch repair. If
Yeah.
too much mutation happens during DNA replication and the cell doesn't catch it right away, and then the DNA is found to be flawed, then apoptosis, programmed cell death, is a good way to, it's a waste of energy, but it's better than having a cell with faulty DNA. Nucleotide excision repair, that is definitely a mechanism for cutting out mutation problems and fixing those. But it happens like when we talk about when DNA gets damaged, it gets cut out and fixed later. Okay.
Yep.
So then proofreading enzymes, we have enzymes that go along and check and see that there is no mismatch repair. So that's happening during replication. And then this last one, adding telomeres to linear chromosome ends, is that impacting mutation, or is
Nope.
that just preventing the DNA from getting shorter?
Getting shorter.
Yeah, so this is the one that doesn't match an impact of mutation. Okay, 23. You know what? We're gonna scroll ahead and look at some mitosis and meiosis stuff for just a few minutes. So, because this is all about mutation. Okay. We've done some matching before. Let's just look at this one. Okay, the centromere is a region in which, so you got to remember what a centromere is and what it does.
The spindle is anchored to the centrosome.
Yes. To the centrosome? No. The spindle, the centrosome is, that's the aster, sorry. The spindle is anchored to the centromere. You're swapping out centromere and centrosome. It's this one. Chromatids remain attached to one another at the centromere. So the place where the two sister chromatids
Oh,
pinch in and they're attached together until anaphase, then
got it.
that point where there's some protein and where the spindle is attached, we say is the kinetochore.
Got it.
And the centromere is the attachment spot for the sister chromatids. Okay, number 30. Where do the microtubules of the spindle originate during mitosis?
Centrosome.
Yes.
Booyah.
Okay, I know. Centrosome, centromere, blah, right? And that's, have the ability to like look into your notes or pull up the slides really quickly if you, because you get to double check. That's the one benefit of maybe online, is that you're getting to double check some things. Okay, what does not happen in mitosis in eukaryotes? So what's the point of mitosis? We're going to replicate cells, but why does a eukaryotic cell want to replicate somatic cells? So you fall and you get scraped, and you have to repair those cells and you have to replace the ones that are missing. So tissue repair or renewal would be a role of somatic cell division to fix damaged cells. If you want to grow from a toddler to an adult, you have to not only have your cells get larger, but do what? Make more cells. So we need development and we also need growth. So development, growth, and tissue repair are all roles of eukaryotic cells.
But mitosis doesn't
my-
account for haploid cells. Meiosis does.
Mitosis can't make haploid cells, so it cannot do reproduction.
Right.
Now, prokaryotic cells split and do asexual reproduction, but they're not technically performing mitosis because they don't have a nucleus. And mitosis is nuclear division, okay?
Mysterious.
So mitosis and meiosis refer specifically to nuclear division in eukaryotic cells, forming daughter cells that are identical for tissue repair, growth, and development, or forming haploid cells that are gametes for sexual reproduction. Mitosis can't do reproduction. All right, 32. So besides dividing uncontrollably, what else could give you a tumor? Having too many cells in one spot.
Lack of apoptosis.
Yeah, if they don't die and get recycled and taken care of, then you could have a whole bunch of cells that aren't obeying the rules in one spot, which is a tumor. You like the very scientific way I said that?
I did. Gave me
All right.
chills.
Okay, progression through the cell cycle is dependent on both extra, so outside, and internal cellular conditions. So cells receive signals from outside, and cells create signals internally. So then now we're having to think about checkpoints and what responds to that condition. All right, so if the DNA has been completely replicated and checked for errors, So when are we
Yeah.
going to be able to check that? We can't check it in G1, all right? We're going to check it in G2. All right.
Me too.
The next one. There is an ample supply of energy and raw materials available. So when are we paying attention to that?
G1.
Yeah. Why even bother replicating the DNA if you don't have enough stuff to go ahead with it? When do we check to see that all chromosomes are attached to their kinetochores?
M.
Uh-huh. Good. And if we see that there is room in the environment and there's no density dependent inhibition, that's an extracellular, an outside thing. When are we going to check for that?
G1.
Yeah. Gotta have space. Okay, good. Oh, does this look familiar?
Oh yeah.
Oh, yeah. All right.
Straight
So
from my nightmares.
I need some comic relief.
Of
Okay.
course.
So we already decided that this was mitosis, right? Remember, because we've got just one round here, and we've got this lining up at the metaphase plate that is just single chromosomes. So, so mitosis for sure. And also notice my meiosis option here is diploid, which is and my meiosis option here says that there are 8 chromosomes. Do you see 8 chromosomes here? Yeah,
No.
I don't see 8 here either. Okay, so how many chromosomes is that? How many chromosomes are here? 1, 2, 3, 4.
Oh.
So it's mitosis, and is it n equals 4 or 2n equals 4?
2n equals 4.
2n equals 4, right? I have a total of 4 chromosomes, and I have 2 sets of 2. So 2 times 2 is 4. And that 1, 2, 3, 4 replicated chromosome set gives it away here. So
Am I to presume that n stands for the nucleus?
N stands for the number of chromosomes. N is just
Makes
number. Yeah. Okay. So now we have to put things in the correct order. So what happens at the end? Which one of these is the end?
4.
Cool.
4. Okay. So these last 3 have 4 at the end. And then we have to decide what comes first, 2 or 3.
3.
Yeah, this was in interphase, right? So 3 is first. So it's between these two. 3, 2. Okay, and then is it 5 and 1 or 1 and 5? So here's 5,
5
and
then
here's
1.
1. Yeah, we line up and then we separate. So 5 and then
Mhm.
1 and then 4. So this guy here. Okay, let's do 2 more, and then do we want you to get to some ones on, oh, let's look at this one, number 40. By the way, if you went back to the video that I post tonight, you could always hit pause when those other questions show up on the screen and you could look at them. If you needed to, they're all there, right? And remember, this is a subset of the questions that you might get because there's more of them and they're just randomly chosen, right? So which of these represents the process of meiosis? We start from 2n, right? So it can't be B. We don't go from haploid to diploid.
D.
That's fertilization, okay? And we end up with how many daughter cells?
4.
4. Okay, so this looks right, okay? And we don't stop partway through because that's just part of meiosis. So we end up from diploid to 4 haploid daughter cells. So we would go with D here. 41. Sister chromatids are separated from each other. And when is that true? Is it meiosis 1? That
Nope.
separates homologous pairs. Is it true for mitosis that sister chromatids are separated? Yes, but is it
Yeah.
true for just mitosis? Or does it
No.
also happen in meiosis too?
Correct.
Yeah, so we would choose the one that's mitosis and meiosis too. Okay. All right. Look at this one here. Spinach cells have a haploid number of 5 chromosomes. I could say this, Spinacia oleracea. How many different ways can spinach homologs be combined? So it's talking about independent assortment. So when I look at that, those chromosomes, I'm going to have 5 pairs, okay? Because the haploid number is 5. So if I have the diploid number would be 5 pairs. And can you see my 5 little X's, my tetrads that I
Yeah.
made? So each of those spots, they could flip-flop. So if I had 2 different pin colors, I'll just circle the one column. So these guys all on the left could be all from dad, and the ones on the right could be all from mom. But what if individually each of them flipped? Okay, so there's how many spaces here? So if one of them flips but none of the other do, you see how you get a whole bunch of different ways? Okay, what is 2 to the 5th?
30?
So 2 times 2 is 4, times 2 is 8, so that's 2 to the 3rd, times 2 is
Oh, 32, sorry.
32, that's 2 to the
Whoops.
4th, times 2 is
2 times 2.
I'm looking at how many different ways can they be combined.
But it's 5,
Am
would
I-
it be 5 to the 5th?
If the haploid number is 5,
Or would it be 10 to the 5th because you have
2n equals 10.
individual halves of each? So technically 10 individuals.
That's where I need my calculator. It's funny where occasionally on the spot my brain goes, oh no, oh no. What if I can't do it for them? I don't know if my little computer one can do it. So there's one there. Okay. There's with 3 spots. So when we look at, okay, 1, 2, 3, 4, it's the 32, right?
Yep.
'Cause
Oh, that's
2
the answer?
1, 2, 3, 4. Yeah, I was writing it down on the paper wrong. So 2 times 2 is 4. So there's 2 of our 2's. 4 times 2 is 8. There's the 3rd 2. Here's the 4th 2. Here's the 5th 2.
I get how it works. I'm just, why is it 2? Why isn't it 5 is my question.
Because
they're pairs?
in every, so in every single spot,
Got it.
so
It's 2
this
pairs
row,
times 5.
it could, well, it's because it could be this way or it could be that way. So there's 2 choices for every row, for every
Yep.
pair, and there's 5 pairs. So 2 times 2 times 2 times 2 times 2, 2 to the 5th. Okay. I don't even know where 1024 comes from.
Yeah, I was wondering about that.
A really wrong answer. Okay. 44, and then I think we'll stop here. If you want to find metaphase 1 of meiosis, what are you going to look for?
Tetrads lined up at the center of the cell.
Absolutely. Okay. And just for, your pausing benefits later, if you feel like scrolling through and answering a few more.
Jesus, so many
scroll.
questions.
Well, I'm just letting you see a bunch of them. So you can look at these guys if you want. Do you want to do this last one
Yeah, I
on
was gonna say,
translation?
can we do that scary one there just for my sake?
Okay, this is a more complex version of a tRNA molecule.
Yeah.
Okay. Now, it says there's a 3' end projecting beyond its 5' end. Do you know what happens here? This is the anticodon down here. Do you know what
Okay.
what's being carried here? You saw it at the very beginning. And if this is the anticodon, then I'm sort of giving it away. The anticodon and the codon complement each other, and this is where it lands into the tRNA binding site and lines
Okay.
up with the mRNA. So what does it bring with it that has to be attached up here? What does tRNA bring with it?
Oh, and protein, right? The things that make the proteins.
An amino acid.
Yeah,
Okay, got it?
yeah.
All right, and we're scrolling all the way through just in case you want to see extra questions and go back through. Okay,
Wait, what's with that last one?
that last one, all right. Nearly all cells in an organism contain identical DNA. So most of the DNA in my somatic cells, barring mutation, it's identical, okay? And each DNA strand may contain hundreds of thousands of individual genes. Think about how I have heart cells and eyeball cells and liver cells and blood cells. Are all of the genes of the DNA that's in each of my cells, because every single cell holds my entire genome, are all of those genes being expressed, turned into proteins in my cell?
At some point, yes, but I wouldn't say simultaneously.
I would say no, because I don't need the genes that code for eyeball color to be working in my red blood cells.
Oh,
Ever.
I see what you're saying. Okay.
Ever. But every single cell contains all the DNA for that organism. It contains the genome. That's why we can swab somebody's cheek cells and come up with their DNA. But their cheek cells aren't going to ever make heart cells. At some point in the embryo, we had special types of tissue that wasn't specialized yet, but cells become specialized by differential expression of their genes. So a cell would never transcribe all its genes within its nucleus simultaneously, because A, it doesn't have energy or the capacity to do that simultaneously. And it will never do it at some point during the cell's life because those cells are differentiated. A red blood cell is only going to be told to make the proteins that a red blood cell needs.
Okay.
And some of those proteins will be identical to other cells, like the way maybe its plasma membrane forms, or to make more mitochondria. But some of those genes are going to be activated only in a red blood cell and never anywhere else, because they're genes specific to proteins that are needed for red blood cell function, like making hemoglobin. Well, do you need oxygen carrying cells that grab it from the lungs and drop it off anywhere else except for in the red blood cells, right? So they're not going to be made other places. So that's why that one is no and no.
Glad I asked about that one.
Yeah, me too. All right, so we're going to